So here we have one of the coolest experiments. This is the Miller-Urey experiment done in the 50s. And basically what they were doing is trying to figure out if we take those molecules we know were on Earth's early surface, if we replicate the conditions, can we get the basic molecules that life needs? So what they did was they got a bunch of molecules together that would have been here, things like methane, CH4, ammonia, NH3, hydrogen, let's see, water, and they basically ran electrical spark, which simulate lightning, which still is very prevalent on Earth. We don't think about too much, but there's a lot of lightning that hits the Earth all around the globe, and it actually creates a lot of fertilizer because it basically converts nitrogen in there, nitrates, which in the fall to Earth help plants grow. So all these molecules that would have been here, and it's all from outer space on the rocks. Then we have boiling water vapor that we would have had from that really hot surface, and then after a few days, organic molecules appeared in the brew. Okay, and basically those would be amino acids. So it just showed that amino acids could spontaneously be created with these raw materials that would have been here on Earth. Okay, so very cool, and it's been replicated afterwards. But there's also, it's really a combination of these molecules, coming from the Earth's early formation, when the large chunks of rock collided to, and coalesced in Earth. And then when our Earth's surface was struck a lot by asteroids over the years. These are giant tubeworms, and they're actually an annelid. Annelida. It's a big worm. There's kind of a tough shell, and this is kind of their blood. They have very unique kind of hemoglobin, and it gets circulated outside their body to pick up, hydrogen sulfide. I'm sorry, that's water, but H2S, and it convert that H2S to oxygen and energy molecules. And you find these called black smokers, but this is very hot water, sulfur. Looks pretty creepy. This is happening thousands of feet down on the bottom of the ocean. Very cool. It hadn't been seen into the 70s, but it's hard to see the scale. But this is a fairly large submersible. Hey, Jackson saying hello. Hey, puppy. That's the pups. Why don't you go relax, okay, dog? Okay? That's the pups. Good dog. Good dog. Okay. So, I'm going to go annotate that. That was a diagram of the worms, and it was, take a look at that diagram. It kind of shows there's bacteria that basically the worm develops from an egg. It floats around, it develops in the egg where it can, and then bacteria start to infect the skin of that worm and basically help. There's symbiotic, which means the bacteria get a home in blood and a place to live. The worm then get this bacteria that can then convert the hydrogen sulfide into oxygen and ATP for it to burn. And they work together, but it's really an infection. Amazing. Right here we have, this is what happens if you put phospholipids in water. You basically, what's going on? There we go. It basically forms my pen is messing up. Basically forms a cell. So hydrophilic head. We see right here, hydrophilic, sorry about the handwriting. And these are right. I can do better than that. Hydrophilic head. Hydrophobic tail. hydrophobic tails. See how the tails are inside, no water, they don't want to be the water, but the heads are on the inside and outside. This is just like a regular cell. So again, what it's showing was demonstrating you can get cells, basically, these structures going if you just have these basic raw materials. It doesn't take some crazy event to make these cell-like structures, these little basically spheres that have an inside and outside. And by the way, this is what our membrane is mostly made out of, like we learned in that last topic. These phospholipid tails, these phospholipids, add some proteins in here. We got a cell controlling the internal environment. And here's ribozyme RNA. It's one of the first enzymes. We need some molecule that can get reactions going and make them go faster. And this ribozyme is a very simple molecule. It's thought that could be one of the first nucleic acids on Earth. And since RNA is simpler than DNA, simpler, it's thought that RNA arose before DNA. But they both carry information. That's the main thing. There needs to be an information carrying molecule in order for organisms to survive and reproduce. And here, this is the endosymbiotic model. Theory. So very well developed. You have different bacteria on Earth. They're developing, and what we think is that at some point you had different bacteria join other larger bacteria and then work as a pair. Okay, so we have our combined cells here basically, and we have specialized. So this would be, if you guess it, this would be an animal cell, and over here would be a plant cell. But with mitochondria and chloroplasts, used to be independent, bacteria, doing their own thing, doing fine, but by cooperating, by being together, just like humans, facilitated success. We work together well, and we help push each other. Think about sitting at home doing all your classwork. It's kind of hard, but you go to school, get stimulated, get pushed by teachers, other students, makes you go further. So this is how, and we know this because there are the mitochondria and chloroplasts have their own DNA, and they can do protein synthesis. And they still, to this day, make their own proteins, that work in the process of photosynthesis and respiration. So really interesting. So we have in each and every one of our cells some bacteria that way back when, I think the thinking is, eukaryotic cells about 1.5 billion years ago. I'll double check that. It's a while since I learned it. That's what I remember. So Francisco Redi did some cool experiments, because people would see meat like this and say, hey, there's maggots and flies popping out of it. So this stuff must be inside meat, and Redi was not so sure. So his experiment, he took the meat and sealed it. And when open container, you got a lot of flies and maggots. But he sealed it up, guess what? No flies or maggots. Whew. Maggots. In the summertime, when your garbage can, if it's been out there in the heat, you see those maggots coming out of there. Whoa! So he also did this other variation where he covered it to keep flies out. And guess what? No maggots. No maggots. No flies. But check maggots, flies when you leave it open. And you've probably heard of Pasteur. but he made these cool things where if you look at this, air can get through here, but it's almost impossible. So air passes, let me get my handwriting checked in here. Air passes, no microbes though, because of the twisty glass. So you boiled it beforehand to kill off anything that was already there. Put it in this flask that's sealed up. 3 days later, no microbes. The air was getting in there, but not any spores or any bacteria or anything. But he boiled it and then snapped this off so that not only air but microbes can get in there. And then you see here, microbes grew. Microbes, yes. Air plus microbes can get in there now.